Coming to the biosynthesis of catecholamines. All catecholamines are derived from phenylalanine. Phenylalanine is an essential amino acid which is obtained from the dietary sources and is hydroxylated on benzene ring to form tyrosine by the enzyme phenylalanine hydroxylase. This requires tetrahydrobiopterin as its cofactor. This biopterin will exactly helps to transfer this OH group to form tyrosine. Tyrosine hydroxylase is found in all cells that synthesize catecholamines and is a mixed function oxidase that uses molecular oxygen and tyrosine as its substrate and tetrahydrobiopterin as its cofactor and catalyzes the addition of a hydroxyl group to the meta position of tyrosine, thus forming DOPA, that is 3,4-dihydroxyphenylalanine. This tyrosine hydroxylase enzyme is a homotetramer. Homotetramer is a protein complex made up of 4 identical subunits which are associated but not covalently bound. This tyrosine hydroxylase reaction is a branch point in the metabolism of tyrosine and is a rate limiting in the catecholamine biosynthesis. Inhibitors of tyrosine hydroxylase, that is alpha methyl tyrosine, effectively lowers the levels of tissue catecholamines and diminishes the amount of transmitter released during nerve stimulation. The next step is the conversion of DOPA, that is 3,4-dihydroxyphenylalanine, in dopamine, which is done in the presence of DOPA decarboxylase. Decarboxylase enzyme results in the removal of carbon dioxide group from DOPA, this carbon dioxide group from the COOH, to form dopamine, which is our first catecholamine. DOPA decarboxylase is a pyridoxine dependent enzyme. DOPA decarboxylase can also decarboxylate 5-hydroxytryptophan, which is the precursor of serotonin, as well as other aromatic amino acids. Hence, it has also been called as aromatic amino acid decarboxylase. DOPA decarboxylase is widely distributed throughout the body, where it is found both in catecholamine and serotonin containing neurons and in non-neuronal tissues such as kidney and blood vessels. In the next step, dopamine is converted into norepinephrine in the presence of dopamine beta hydroxylase, and this norepinephrine is the second catecholamine. Like tyrosine hydroxylase, this dopamine beta hydroxylase is a mixed function oxidase that uses molecular oxygen to form the hydroxyl group added to the beta position or the beta carbon on the side chain of the dopamine. Ascorbate reduced to dihydroascorbate during the reaction and provides a source of electrons. Dopamine beta hydroxylase is a tetrameric glycoprotein containing copper, that is Cu2 plus, and this Cu2 plus is involved in electron transfer in the reaction. So it is important to note here that copper chelators such as diethyl dihydrocarbamate are the potent inhibitor of this enzyme. In the final step of the pathway, the norepinephrine is converted to epinephrine. In the presence of enzyme phenylethanolamine N-methyl transferase, it transfers a methyl group from S-adenosyl methionine to the nitrogen of the norepinephrine, forming a secondary amine, that is epinephrine, and this epinephrine is the third catecholamine.